Yeah, it
Yeah,
looks good.
that looks
That
good.
looks good. Okay.
Okay, cool. Thank you.
Mhm.
Yeah, thank you again for the plug. We've had really great webinars and just great partner engagement over this month and learning about invasive species and all the different work that's happening. Today, I'll be talking to you about a shared path forward. I'll be giving some updates on our Hawaii Interagency Biosecurity Plan and also the new HISC-CGATS 2025 joint strategy, which I know a lot of people that are in attendance today had a lot of help and input through both those planning processes. So again, thank you, Rashawn. My name's Chelsea. Again, I'm a planner with the Hawaii Invasive Species Council. Just for folks that are unfamiliar with the Hawaii Invasive Species Council, so our council is made up of the 6 state departments that have some type of jurisdiction in invasive species prevention, control, research, or outreach. So that includes Department of Land and Natural Resources and Department of Ag, which are kind of the 2 major entities dealing with invasive species. And again, the 2 chairs of those departments are co-chairs of our council. And we also have members from Department of Health, the Department of Business and Economic Development and Tourism, Department of Transportation, and the University of Hawaii. And the council comes together and helps just push through policy directives on invasive species. We also have the Hawaii Invasive Species Council funding that goes through and helps support a lot of our partner projects that are working on invasive species issues. Okay, I know I'm not starting it off great with all these words right now, but this is a timeline we put together during the planning process of our 5-year strategy with HISC and CGAPS. And I thought it was really great that we put this together, and we weren't really able to share it during our workshop in August where we brought all our partners together. And I thought this was a great place to kind of just, before we get to where we are and where we're going, let's reflect on where we've been. And this timeline is milestones of kind of some of the most damaging invasive species introductions. I mean, we could go back further. I mean, we're starting here in the 1770s with the introduction of feral ungulates. 1826, mosquitoes are introduced. But I also really wanted to highlight the collaborations and the partnerships that have occurred over this course of time here in Hawaii to address these invasive species issues. In 1850, botanists came together to really focus on, how can we control feral animals in the areas? And then shortly thereafter, not shortly thereafter, actually fast forward, we have the first watershed partnership starting up in East Maui in 1991. 1995, the Coordinating Group on Alien Pest Species is formed. 1997, we have our first Invasive Species Committee on Maui formed. And then after that, the subsequent counties start having invasive species committees. In 2003, the Hawaii Invasive Species Council is established. 2005, we have the Division of Aquatic Resources starts their Aquatic Invasive Species team. And then in 2016, the Mamalo Poi Poi Airports program starts up. And after that, we have some really great initiatives that come about. The Hawaii Interagency Biosecurity Plan. Governor Ige chairs the Western Governors Association and launches the Biosecurity and Invasive Species Initiative. And of course, this is not a comprehensive list at all, but it just really shows the partnerships and collaboration that are really at the heart of invasive species management here in Hawaii. And I think we just do a really great job of that in Hawaii. And that's kind of what I'm pulling together for this presentation and the updates about these plans. I just wanted to highlight, some of the biosecurity plans that have come about. 2015, there was the Regional Biosecurity Plan for Micronesia in Hawaii. And shortly thereafter, we had our Hawaii Interagency Biosecurity Plan, which I'll be talking about. And we had a few island-specific biosecurity plans. And New Zealand's awesome, but their biosecurity plan came after our biosecurity plan. So we were ahead of the game, but still they launched their biosecurity 2025. And then, like I said earlier, Governor Ige launched that Biosecurity and Invasive Species Initiative with the Western Governors Association, which brought a lot of attention to invasive species in our biosecurity plan here in the Pacific. It begs to question, is biosecurity trending? I looked this up and there's almost 17,000 posts with the hashtag biosecurity. But Ariana Grande has, like 40 million. So, I mean, I would say no, it's not trending, but hey, it always could be. So that's my little joke plug for now. And now we'll get into the fun numbers. Okay, so the Hawaii Interagency Biosecurity Plan, this was released in 2017. It's comprehensive in scope. So it really looked at the full picture of biosecurity here in Hawaii. It focused on pre-border, border, and post-border actions that are needed, and also public awareness and support needs. Its collaborative and approach. Again, it brought together those 6 state departments, also stakeholders from various sectors, and our other partnerships and organizations that are working on invasive species through a year-long planning process. And their input really brought us to these 147 action items that are laid out in the plan to address the gaps and weaknesses in the system. And I mean, that's a lot of actions, but this is laid out over the course of a 10-year path forward. So just kind of focusing in on what does the biosecurity plan address for the folks that don't know. Again, it's focusing on these different layers of biosecurity. So pre-border, border, post-border, also public awareness and support. And within each of these levels of biosecurity, there are policy actions, process or programmatic actions, technology, infrastructure, funding, or staffing. So if you actually go in and look at the biosecurity plan and you look at the appendix of the 147 actions, they'll be abbreviated with like a pre-poll. So pre-poll is a pre-border policy action, some type of legislation that needs to happen offshore to address that issue. So just kind of, if you do look at the biosecurity plan, that's a little bit helpful to know what those abbreviations mean. Starting in 2017, this is kind of what we were looking at. These were some of the major gaps that we wanted to address. When we're looking at funding, there's a lot of soft funding, especially for those operations that are on the ground controlling invasive species, like our invasive species committees or watershed partnerships, kind of those front lines for the post border. There's experiencing shrinking federal grants. And then the 2 major agencies, Department of Agriculture and DLNR, that have the biggest mandate with invasive species management, they only receive 1.4% of the overall state budget. Staffing, 2008, 2009, there was a huge economic downturn, and a lot of these agencies took a major impact, including Department of Agriculture. And it's just been a struggle getting numbers back up to those pre-2008 numbers. And those are our front lines. Those are the people doing inspections at the ports of entry and making sure invasive species aren't coming into Hawaii and establishing. Infrastructure needs, so inspection facilities at those ports of entry, places where you can have containment and be able to control what's going on and what you're looking for. Also, the need to update our current biocontrol facilities with Department of Agriculture and the USDA. Policy needs, addressing ballast water and biofouling, also inter-island nursery certifications and inspections, and then the need to inspect non-agricultural commodities. Right now, Department of Agriculture can only look through commodities that are agricultural or whatever that listing is. And it would be great if we could expand that to include non-agricultural commodities. And informational, so we need to know where to look. We need risk assessments to identify those high-risk pathways and also those species that are kind of on the horizon and we're at risk of being introduced. And, invasive species, once they get here, they cost a lot of money. And I'm sure a lot of you have seen this slide. Josh uses this for the updates. And I think it's just a really impactful slide to show, if brown tree snake got here, it would be $2 billion per year in damages, 200 million per year on Hawaii Island alone for little fire ant. 700 million a year in management of Myconia calvescens. And then the two newer species to Hawaii that we don't really know those economic costs of rapid ohia death and coconut rhinoceros beetle, but definitely these are having an economic impact. And looking at the cost and context, right? This is implementation of the biosecurity plan on a yearly basis. So 38 million per year to it over a 10-year path if we do everything in the biosecurity plan. And that compared to the introduction of brown tree snake here. I mean, that's just a drop in the bucket. It's really small numbers compared to the damages and costs that each of these species could have. Something like red important fire ant that we don't have here in Hawaii, but definitely could be on the horizon, or little fire ants spreading throughout the state. So what are we looking to? Where's the light in 2027? I mean, this is where we're trying to go. And these are just a few of the major um, actions we would really like to see achieved at the end of this 10-year plan. Risk assessments and electronic manifesting. Department of Agriculture is already starting training on their electronic manifesting system. Regulation of ballast water and biofouling inspection facilities, doubling up inspection staff for both imports and inter-island shipments, and then also increasing field capacity at Department of Land and Natural Resources, people on the ground controlling invasive species, and Department of Health, the vector control branch, which manages a lot of those mosquito outbreaks, rat lungworm, that kind of stuff. Okay, now we're into where we're at. So 4 years into implementation, we're 65% of the way to completion. I mean, that looks really good. And when you break that number down, we're looking at 34% of those actions are either completed or they're ongoing in perpetuity. 31% of that, they're in progress towards completion. And that could be a long road, but at least it's been started. And then we have 35% that have not been started at all yet. And those are kind of, I mean, we'll get into it when I go through these updates, but they're harder things to push through. So looking at our accomplishments so far, completed within the last, oh man, I think this was just last year, but it's so hard to tell time nowadays. But last year, the Board of Agriculture approved a domestic restriction on the import of all species in the myrtle family into Hawaii. And this is to protect our own native myrtles like ohia from pests and diseases that can be carried on those plants. So such a huge accomplishment. Department of Ag has been hosting post-incidental meetings for early detection and rapid response to little fire ant on Oahu. Kind of some past things, 643.org is an online pest reporting tool and app that's been released. That's fully functioning and people are using it and we've been promoting it. And it's just a great tool that the public can use for reporting pests. Extension agent positions for UHC-TAR. And then I'll move into some things that are in progress. And so we have been a multi-agency biocontrol working group that has been specifically working on the task of a regional biocontrol facility and looking at talking about facility design, how to get funding, and also communications and outreach on this topic. So those are ongoing monthly discussions. Also working on something that complements that domestic restriction on Myrtaceae, looking at the federal side. So this would be a federal restriction on the import of all species in the myrtle family. So restricting the import of things from foreign sources. And again, this is gonna help protect our native myrtle species like ohia from any pests or diseases that can be carried on those plants. The Division of Aquatic Resources has tested out molecular techniques to identify aquatic invasive species, which is great for being able to identify high risk and invasive species. The University of Hawaii CTAR is partnering with Department of Ag in developing diagnostic tools for inspections. So a really great partnership there. But again, we have a lot of needs we need to address. And these are kind of the harder ones that they deal with funding and positions and policy that can be a little bit of a longer road. So those include the capacity to co-manage vessel biofouling and ballast water discharge. We need a team that can enforce regulations on those, and getting aquaculture extension agents. Roshan, you can say if this is wrong, but I don't think there is anybody currently that is an aquaculture agent for CTAR, and that's a huge deficit there. A biosecurity emergency response fund. This would be something put to the side for when new pest incursions happen, and we wouldn't have to tap into existing organizations budgets or resources, and we could tap into emergency response fund. But again, in the time of COVID and what we're dealing with right now, we're really dealing with maintaining capacity and keeping the positions and not going back to where we were in 2008, 2009, where a lot of our inspectors and our resources for invasive species management were cut back. Okay, we're just planning away here. So that brings me to the HISC and CGAP's 2025 joint strategy that was just released last year. And a lot of you were part of that planning process. And it was, again, over the course of a year, we used partner input on invasive species priorities. And they helped us identify what the priorities should be over the next 5 years. And again, this is done under the guidance of the Hawaii Interagency Biosecurity Plan. Nothing that you see in our 5-year plan isn't already addressed in the biosecurity plan. But what it does, it takes 10 strategies were identified, and it kind of creates a roadmap. So we were able to get a little bit more detail in how we can achieve these strategies and get to the end goal by identifying key actions with our partner network. And I just have this up here because it was one of the interesting things that happened in the planning process of putting together this joint strategy. And I guess I should say, so I've been saying this so much, but Hawaii Invasive Species Council and the Coordinating Group on Alien Pest Species, we share the same vision, which you see above here. We have the same vision, but the way we accomplish it is a little bit different. And it was interesting because a lot of our partners that have been working with us for a long time didn't exactly know what the difference between our agencies were. But those differences really complement each other. And I think we're able to pull our networks together and really achieve a lot. And that's why we came together in this 5-year strategy. It just made sense to come together and have a shared vision and a shared strategy for the next 5 years. And maybe this will be how we do it ever more. So these are the 10 strategies that were developed through a year-long process with our partners and just fine-tuning it with our planning team. And you'll see, these are broad sweeping strategies, but when you break it down and look at the plan, there are some specific key actions to help us accomplish these goals. So prevention and early detection, rapid response for new terrestrial invasions. So these are things that haven't shown up in Hawaii yet. And then the second strategy gets into intra-island movement of terrestrial invasive species. So things that are already here and keeping them from moving across our islands and across our specific islands. Aquatic biosecurity, large-scale control of high-impact invasive species, working towards that Pacific Regional Biocontrol Center, increasing and diversifying funding for invasive species priorities, engaging in supportive communities. And so the last 3 are organizational development goals. These are specific to CGAPs and HISC. For HISC, we're looking at more engagement across our sectors. So, a lot of people said, you're a little too natural resource focused. Look at agriculture, look at health, let's spread it out a little bit more. So that's what that goal focuses on. And then a strong CGAPs network and stable CGAPs funding and staffing. So these are the goals, and I won't go into the key actions because here you can help because again, this is a plan made from our network, and it's not a plan, just a plan, it's a plan for action. And that's what we're doing with our working groups. These working groups are our strategy teams, and we've restructured some of the Hawaii Invasive Species Council working groups to be the implementation teams for these strategies. So what we have now is a prevention and EDRR working group. This is co-chaired by and Layla's on here, so please correct me if I'm wrong, because we're going through a little shift in chairs. But Mike Melzer is co-chairing along with a representative from Department of Ag. Kevin Hoffman was the chair, but he's since retired. So we're looking at somebody to replace that one, and they're working on that strategy 1 and 2. Outreach, again, this is continuing on with Christy Martin, and this is taking on the community engagement strategy. Aquatic biosecurity, this is a totally new working group, and it's already going really well. We have had, I think, 3 meetings, and it's co-chaired by Natalie Dunn and Kim Fuller from the Division of Aquatic Resources. And then, like I said earlier, we do have a biocontrol working group that is meeting monthly and really focusing on that regional biocontrol facility. In the works, we have the large scale control and our funding strategy that we're not sure if a working group is the best place for that. And there are already projects that are doing large scale control. So we're kind of reaching out to individuals that are involved in those groups and just seeing, is there a good forum or a place you want to have a conversation about the challenges you might be experiencing in large scale control? Maybe it's permitting or outreach or something. So we'll be kind of figuring out how to work on that. And I'm totally happy to hear from other partners if you have any ideas how to move those actions forward. Well, thank you for listening. That's the end of my talk, and I really appreciate everybody sitting through and learning about our plans. But again, these are plans for action, and hashtag biosecurity. We can make it really cool and trendy. And you can reach out to me if you would like to join any of the working groups. Feel free to email me. You can also visit our website, and you can subscribe that way also. So thank you, Rashawn. I will stop sharing.
Thanks, Chelsea. Any question for Chelsea? So Chelsea, I have a question. So CGAPS has also a strategic biosecurity plan at the beginning?
No, actually, CGAPS has definitely been involved in the Hawaii Interagency Biosecurity Plan. They were part of that planning process. And what they've had is they have a 5-year strategy, and basically the CGAPS and HISC strategies needed to be updated at the same time. And we just thought, okay, we would share the planning process and then develop our own plans at the end. But as we work through this planning process, it just made sense to keep them together and have this joint strategy together.
Okay, thank you. So I saw there is a biocontrol facility. Yeah, is that been in process of building or it's already there?
Oh, I wish. It's a long road to do that. We are making headway with identifying different pots of funding that could possibly support planning and eventual construction costs. We're looking at federal funding and putting in appropriations requests with our partners that way. But right now, we are just in the beginning phases of trying to get money for a feasibility assessment. So currently, we're identifying potential sites and who we could partner with in order to put those facilities together, who would be in those facilities, and getting like MOAs together of what the roles of the different agencies would be. And hopefully, with it soon, we'll have some funding to start the process of planning and construction.
So questions for Chelsea. So, okay, if there is no questions for Chelsea, thank you again, Chelsea. And we will go ahead with Sienna Sim's presentation. So Sienna, So Sienna is a research biologist at Pacific Basin Agricultural Research Center at Hilo, USDA, ARS, and she basically does research using research and pest management using molecular tools. And today he will be talking about applications of next generation technology to Queensland longhorn beetle management. Sienna.
Hi, thank you so much for that introduction, and thank you for inviting me to speak today. I'm really excited to share with you some of the new work that we have done on the Queensland longhorn beetle. So just to clarify, you can see the presentation part, not the future slides part, right? Can I get a shocker from someone? I'm going to assume you guys can see the right one.
Yep.
So this is our center. I'm obviously not there right now, but because I'm working from home like everyone else, but this is one of about 100 centers for the USDA Agricultural Research Service around the United States. And our task is to conduct research to develop and transfer solutions to agricultural problems. So the study systems that I employ in my research program are predominantly to fruit flies. And more recently, this species right here, it's a Queensland longhorn beetle. And we use these study systems to, one, expand on existing control methods, such as the sterile insect technique, but also we're all trying to develop new control methods for species that we don't have control methods for management programs. So today I'm going to be talking about what we're doing to help solve this Queensland longhorn beetle problem and ways that we can prevent its movement around this island, but also hopefully prevent its movement to other islands, which is also very important. So this beetle is an insect, it's a beetle, and it's in the family Cerambycidae. So it's a longhorn in the subfamily Lamianae. And it has two closely related relatives that are actually really infamous and causing lots of problems on the mainland. Anaplophora glabripennis, which is the Asian longhorn beetle. It infests hardwood trees like poplars on the mainland, mostly in the northeast and a little bit in the Midwest. And then there's Anaplophora chinesis, which is a citrus longhorn beetle. As its name suggests, you can imagine it infests citrus trees. So if it became more established in largely citrus growing regions like California, Florida, and Texas, that would also become a real problem. So the species we work on here is Acalylepta aesthetica, the Queensland longhorn beetle. It's known because it's an insect, obviously. It's ectothermic, has really long antennae that aren't horns, they're antennae. It's what they use to interact with the chemical world. Their larvae are wood borers, which is why they can become problematic. And this particular species is invasive. Not all Cerambycids are invasive. There are some native and not problematic species of Cerambycid established and present here on Hawaii Island, but this one is not one of them. It's one of the bad ones. Because they're invasive, and they can be invasive, and their larvae are obscure, they can become major pests without you necessarily knowing that they've even arrived. And this is because of their life cycle. So like all other endopterygotes, it goes through a complete life cycle. Starts off as an adult, which lays eggs. Those eggs emerge into larvae, which then grow and grow and grow until it decides it's ready to pupate, which case it does. And after it's pupated and ready to emerge as an adult, it does that. However, because most of the life cycle actually occurs inside of a tree, these are species that are notoriously easy to move around in wood packing materials, furniture, stuff like that. What we know is that prior to its arrival to Hawaii, it was endemic to Australia. And before it was found in Hawaii, it was actually only 4 documented finds were documented. 2 were in Queensland and 2 were in New South Wales. And so a little brief history of this species in Hawaii. It was first detected on Hawaii Island in the Pahoa area in 2009, and later detected again with multiple adults were found in Lower Puna. The following year, it was first documented, its first host was documented, which is the breadfruit. So before that, we didn't have any idea what it was eating. And then in 2018, it was detected as causing damages mainly to cacao. That's coincidentally when I also started my job. And since then, we've done some research involving a citizen science project, an active beetle tracking project, and some genetic analysis, which I'll elaborate on in the next few minutes. So now that it's here in Hawaii, we know that it has several confirmed hosts, the breadfruit, cacao, citrus, various citrus species, and kukui, which although it doesn't, it's not necessarily an agricultural crop, it does have a lot of cultural importance. There are whole lists of lots of other things that you can infest. Some of these are not necessarily preferred hosts, but they can eat it, which kind of also demonstrates how broad of a host breadth this has. Some signs of damage include abnormal oozing in your tree. So if you see any of that, you might have beetles. Girdling, which can cause limb or tree death. And these kind of dime-sized holes, which is what the adults emerge out of from the tree that are really conspicuous. So we're going to try to use genomics and population genetic analysis to answer a few questions. One, we know that it arrived, or it's originally from Australia, but is that actually how it got here? Or was there an intermediate location that we actually received the beetles from? Because to my knowledge, we don't import wood products from Hawaii or Australia. And so it could be that we got that some secondary location sent beetles to us and they got beetles from Australia previous to that. And how many times did it get here? Did it arrive the one time or multiple times? And this is one of the questions we can answer with population genetics. Now that it's here, we want to know how widely it's distributed. Has it gone as far as it can every generation since it's been here, or does it mainly stick to the same location? Normally what you would do to answer these questions is use lures and traps to see, where the beetles are found when you put your trap out. Unfortunately, there are no species-specific lures for this species currently, and cerambycid lures that have been tested previously showed in 2 different trapping experiments that tested the attractiveness of these cerambycid pheromones. They didn't really work. It turns out these particular beetles, Queens of Longhorn beetles, not interested in these cerambycid pheromones at all. This is something that represents a gap in this species. And so I'm working with 2 chemical ecologists, Damon Crook with USDA APHIS and Dong Cha with USDA ARS, who are going to work at isolating these pheromones for this for QLB that can hopefully be used for trapping and monitoring for early detection and rapid response in the future. So another way that we're answering this question is actually using citizen science. This it accomplishes 2 things. One, it tells the general population, there's this beetle, don't move wood around. And also it enables us to communicate with the public in a way that if they find a beetle, they can bring it to us, and then we can either use it for genetic analysis or to introduce into our colony. But also it tells us where the beetles are. So so if you go to this QR code, you'll end up at this ArcGIS web app that anyone can access either via your mobile phone or your web browser. And that'll enable us to document the date and location that people find beetles, and also document the host species that those beetles were near or found on. And so if you go to that QR code, you end up at this little form, we get some information, and then we can contact you if we want to investigate further. And you also attached a photo with your find, and that's how I've been using, what I've been using to verify some of these finds. So I got responses from all over the island, and luckily of the people who sent photos, the only places where there was a positive find of the Queens Longhorn beetle is in this area in East Hawaii, pretty close to where it was originally found, and which is great because hasn't yet been detected in the Hamakua Coast where we grow a lot of cacao, or in Kona where a lot of citrus is grown. So if we zoom in to where these positive finds were, we see that a lot of it is in Hawaii Paradise Park in the Kei'au area, and Mountain View and Orchid Land. And there are these two beetles that we actually saw, or that were detected in Hilo. And I don't know how many of you are familiar with where the geography of Hilo, or what the different landmarks here are, but right here next to the airport is the transfer station. And so my hypothesis for this is that people were bringing, or had brought infested wood or some sort of infested green waste to the green waste facility here. And at some point, those beetles either hop out or emerge, and then were found kind of en route. And so this is represents a kind of new information that we got using citizen science that we wouldn't have known otherwise, because we didn't have traps there and we didn't know that that was somewhere we should be looking. So now that we know kind of how broadly these beetles are being distributed across the island, I'm also starting this new project using harmonic radar for beetle tracking. And as much as I wish that I was training my dog to identify or sniff out beetles, she's not quite trained for that. So we're using this RECO unit. It's a transceiver unit that people use to find people who've been buried in an avalanche. So the way it works is this RECO detector receiver emits a directional radar signal, and that's kind of like the beam of a flashlight, and you wave that across your landscape after you have released a beetle that has a little reflector on it. When the excuse me, when the radar signal hits a RECO reflector that you've attached to a beetle, it's echoed back to the detector and then points you in the direction of that beetle. The closer you get, the louder that beep is, and until you find your target. And he's right there. Say hello to Mr. Beetle. This is something that I'm working with Dr. Matthew Siderhurst from Eastern Mennonite University, who came here and visited and brought this technology. And we decided, you know what, QLB would be a really great system to test this out. So this is kind of what we're attaching to the beetles. It's a little reflector unit. This is that black square or black cube, and then a metal antenna that we're using. It's about 2 shakas long. So it's pretty long because it's supposed to reflect the wavelength, or some proportion of the wavelength that's being emitted and received by the reflector unit. And so this is actually not the way that we eventually attach the reflector to the beetle. We did it like this. So the antenna are parallel to the body instead of perpendicular. I thought perpendicular would work because it has such long antenna that are about the length of these of the reflector antenna. But no, we attached it the 2 different ways. We set it loose in a flight tunnel, and it was really cranky with it perpendicular, but with it parallel, it flew just fine. And so this is the design, or the placement that we had for the beetles that we use in our pilot study. So what we did was we released 5 beetles in a heavily infested kukui site at Kamehameha schools here nearby in Kea. And this was a joint effort between our center, Eastern Mennonite University, and Hawaii Department of Ag. Over 11 days, what we did was we tracked 5 beetles, 4 of which we actually tagged on the back of the truck in the middle of the night. So that was exciting because they do bite. And over the course of 11 days, we repeatedly found some of the beetles in all sorts of different places. And so these are just a handful of the photos that we have. It was found on weeds, a lot of it found in the leaf litter, on the trunks and our branches of the trees, and sometimes in the canopy. So now we're going to go spend I'm going to take you through 11 days in the life of one of the beetles that we released. This is the one that we were able to track for the longest period of time. It's going to be really exciting. Watch, you'll see. So it started in a tree or on a tree. The next day was found in the canopy. Later that day was found on a leaf on the ground. Then it was found again in the canopy, on a tree trunk, dead leaf on a branch, a palm frond, a Hawaiian tea leaf, and then a leaf on the ground. And there's a line here because this is the only actual flight we witnessed. It was first found, we got the signal that it was on this one tea plant. Then in trying to find it, so we could take a picture, it flew away and then landed about 30 meters. So it was able to fly about 30 meters in one jump or one flight. So it didn't seem like the antenna was impeding its movement. The following day, it was found on a branch in the canopy. And the last time we saw this beetle, it was on the grass. It wasn't doing too well, and we tried to pick it up, but it didn't want to come with us. And that was the last time we saw him. I think he might have been eaten by pigs, because there was a lot of pig activity there. And since he was on the ground, I mean, that's exactly where a pig would probably find him. So what does this tell us? So, I should first say this is about maybe the length of a football field, 100 meters long. This tells us that even though they can potentially move like 30 meters at a time, they don't disperse very much. They have very low dispersal. According to the beetles that we follow, they spend a lot of the time in the canopy and then in the leaf litter. So they kind of go down and then up and then up and then down. They're highly camouflaged because they're like, brownie mixed stick face. They just look entirely like a stick or the dirt or some sort of leaf. I was staring at a patch for, I don't know, a good minute or two, knowing that the beetle was there because that's where the signal was telling us. And I wasn't able to find it right away because they are very, very highly camouflaged. And because these reflector units aren't unique, so one sounds the same as the other, because we released 5 in one area, it was difficult without marking the beetles to find out who was who. So we might want to limit the number of individuals that we tag we might want to limit the number of individuals we release in one area to 1 or 2. Let's see. So some other information that we have is, so we did a small genetic kind of diversity experiment and also made a neighbor joining network comparing it to other cerambycids on this island. So there's 2 cerambycids here, and there's actually a more updated network that I don't have, but it's pretty distinct genetically at the mitochondrial Co1 locus from other beetles on this island. And also other invasive beetles. So this is what we're using to develop a diagnostic tool. So if we get some frass or some other non-destructive part or some life stage that's difficult to identify, we can sequence it and know definitively if it is Acalyptus thetica. Another thing that we know about this beetle on this island is that the genetic diversity is very low. It's actually non-existent. And so at the Co1 locus, there's absolutely 0 diversity for all the individuals that we sampled. And this tells me that as far as how it got here or how many times it got here, it likely only arrived once, and it was one piece of infested wood that had a boatload of one female's offspring that has now become its own or that's now established here. One other thing that we have identified is its susceptibility to Bavaria infection in the wild. So this is not a beetle that we treated. We found this beetle, or someone gave it to us, and we wanted to include it into our colony. Unfortunately, the beetle kicked the bucket shortly after and developed these, fungal things on its body. We sequenced it and saw that it is in fact Bavaria. So we're working with a company to include QLB, cacao, and kukui onto their labels. So that way cacao growers can spray this Bavaria product onto their trees, and then they won't have to and so it'll be represent a labeled use of that product. Let's see. So until then, I think what we're recommending right now is for people to survey, sanitize, and report if they find that they have beetles. This is current. Oh, and also based on what we know from the tracking data, I think what we would also recommend is not to move leaf litter kind of around infested areas around, because that is maybe where they might hang out. And if they were to spray some sort of insecticide or pesticide, we would recommend spraying the canopy, because that's where they hang out a lot of the time. This is my technician, Kina, who's spending a lot of time as a beetle husbandry forestry person. She's taking a lot of care of our colony, and that colony is what will be used for testing Bavaria kind of more explicitly, but also other insecticides that might be susceptible, or that the QLB might be susceptible to. All right, so in conclusion, to answer some of the questions, we use citizen science because in the absence of a really strong or good lure for detecting where beetles are. This was used to determine that beetles are mostly distributed in the east side of this island. We use harmonic radar and active tracking to see how much a beetle can disperse within a day. And this is something that we want to expand on in the future. And also we did a genetic analysis that shows that the beetles here are very distinct. We can develop diagnostic markers for them and differentiate them genetically from other species. And also we're working with a company to get QLB and cacao and kukui on their labels, so that way people could start using that. In the future, we're going to, like I said, test the efficacy of other insecticides, conduct a bigger active tracking study, and then use that information to helpfully inform others on how to do sanitation and pesticide application. And then test our QLB specific markers so that way we could use them for diagnostic purposes. All right, so this was a very much a group effort, and I have a lot of people to thank that did a lot of the work that I just presented here. And funding sources include HISC and internal funding by the USDA. All right, I'm now ready for questions.
Thanks, Sienna. Let's see, questions for Sienna. I can see many questions in the chat box. Fred has a question.
Sorry, I don't know where they start.
So Stephanie Joy has a question. So she was asking, the reflector technology is very interesting. They need to track some other snail movements or native snail movement. Could you be able to provide some information or link for the product?
Oh yeah, absolutely. So yeah, I can connect you with those people. So it's a Swedish company that sells this this unit, and then they also sell little reflectors. You can, you'll have to make the little tags yourself. I think, how big are your snails?
Stephanie, you need.
Yeah, hi. Sorry.
Stephanie, you might be in the mute. Okay. Stephanie? No. So anyway, we go ahead, and I think Fanny has answered Fred's question, and Marshall, Diane,
yeah,
Marshall.
Sorry about that.
Okay, Stephanie. Yes.
Yes, sorry. We're using we're looking at Euglena snail movements and in regards to also our native snail, Hecatonella, that kind of thing. So yes, we'd be interested. Yeah.
Yeah, if you send me an email or something, I think my email's in the contact list somewhere. Yeah, okay. I can put you in contact with them. The reflectors themselves are very, very small. And if your snail can handle a very long antenna, you can actually have pretty far reach. So, and considering they're snails, I'm hoping they're kind of slow. So that's also in your favor.
Well, yeah, you'd think so.
Yeah. Well, they're not going to take flight and then be, 50 meters or, 100 meters away in one go. So yeah, I think the fact that they're terrestrial, that's definitely in your favor. But yeah, I can send you some of that information for sure. Oh, wonderful. And sorry. So someone also asked if I'm adding, we are adding breadfruit. Yeah, breadfruit was one of the species that we're adding. We're trying to get on that label amendment with that company. But yes, good question. I forgot. But yes, breadfruit's included.
Oh, and actually one more thing, since we're dealing with the coconut rhinoceros beetle, it seems like those folks, there's always been this question about how far they travel to reach a trap. So it'd be great for that. Anyway.
Yeah, that's a really good point. So my collaborator, Matt Siderhurst, it went to Guam and did this with the coconut rhinoceros beetle. And so I know I know that there's some information on that. They're quite a bit tankier than than our beetles. So I'm sure that they have a better or greater capacity than, and I don't know, I like how actually, yeah, there's all sorts of questions that you could ask. But so I can also put you in contact with Matt Siderhurst, who's the one who brought this technology to our lab.
Yeah, that'd be great, because I'm like the forest pest person for the Army. So yeah, I'm dealing with those too. Yeah.
Yeah. Great. Thank you.
Yep.
For this species, there are no currently approved pesticides. If you, I know someone who's used, I think, Admire Pro when their cacao were young. So because they weren't going to be selling, bearing fruit or creating, selling any of those products at any time soon past like after that the pesticide has, done its thing. I think they were able to use use that for not the right, sorry, I'm stumbling a bit, but they were able to use an off-label use of that product. I don't know if that makes any sense. And someone who knows a lot more about pesticides should definitely chime in here if that's actually allowed. But they did use it before they were the fruit bearing.
So Sienna, I think you have lots of questions. Can you address those questions in the chat box after this presentation? And let's give thank you. So welcome back again. So our next speaker is Michelle Au, right? Au? Ooh. How do I call your last name? Ooh?
Au.
Okay, Michelle Au. She is a graduate student at Department of Plant Environmental Protection Sciences with Dr. Mark Wright. And she will be talking about a new pest. It's a Rami moth. It's a pest of fire crops, I think, and it's attacking our native plant on Big Island recently and on Maui before, yeah?
Yes.
So yeah, Michelle, whenever you are ready, please.
Thank you very much, Roshan, for having me today. My name is Michelle. I am a graduate student in the Plant and Environmental Protection Sciences Department at UH Manoa. And today I'm be talking a little bit about a project I've been working on. It's a new pest advisory for the Rami moth, Arcticurula in Hawaii. It was first detected in Maui and most recently in Hawaii Island. Okay, so a little bit of background. The Rami moth, Arcticurula, is a Lepidoptera in the Noctuidae family. Noctuids are a fairly large group of both large and small moths. The Rami moth is native to Southeast Asia, and it has since spread up north into Japan, westward into India, Sri Lanka, and then southeast into Oceania, including Australia, Fiji, and Papua New Guinea. The Rami moth feeds on a range of host plants in the nettle family Urticaceae, primarily Bomarea species. There are several different species of Rami in different parts of Asia, and Rami is grown primarily as a important natural fiber crop in places like China, India, Southeast Asia, and Pacific Rim countries, and that's kind of where this moth has spread into these areas. So its introduction into Hawaii, it was first discovered on Maui in November of 2018 by DLNR Division of Forestry and Wildlife. The larvae were seen defoliating mamaki in Olovalu in the West Maui Mountains. And then within a week, the same caterpillars were found feeding on mamaki plants in the Olinda Rare Plant Facility in East Maui. And because of the timeline and the location, we believe that the Rami moth has been here for a little bit, and we see populations starting to pop up. And this is the first record of the Rami moth in Hawaii as well as the United States. So the Rami moth poses a threat to our endemic Kamehameha butterfly populations because it's competing for the same native host plant resources. It's also damaging to our native forests, especially those that have a lot of native endemic Urticaceae plants, especially forests with a lot of mamaki understory. So this is a really good image that was captured in Olovalu by DLNR. You can see the Kamehameha butterfly larva on the top in the blue, and it's on the same plant as the Rami moths. And you can see huge damages of defoliation of the mamaki. This is also damaging and has potential impacts on our agricultural industry because we do have an emerging mamaki tea industry with farms on both Maui and Hawaii Island. So in the next few slides, I'm just going to go over a little bit of description on the different life stages, things to look out for when you're in the field, if you grow mamaki, anything like that, things to spot so that if you do detect this pest to alert us so we can do proper surveys. So the eggs are clear white in color. They're circular in shape, about 1 millimeter in diameter, and laid singly on the underside of leaves. In early instars, they're green and black in color, but then as they develop, they become this vibrant yellow and black in coloration. And there is some variation in their colors depending on how much yellow and black the larvae portray. They do have these distinctive red and orange spots on the side of their bodies. And they also have these hair-like spines that protrude from their body. The pupae, when they're ready to pupate, will tie the leaves together or other debris near the tree or in the soil surface. And it takes about 25 days for them to emerge. The adults are fairly large moths. They can be mistaken for the black witch, which is a larger, well, not noctuid anymore, a rebid. And so they also have that dark head capsule and black, dark brown forewings. But for the Rami moth, they do have these speckles of silver and blue on their forewings, and then these very distinctive silvery bluish markings on their hind wings. And then in terms of behavior, the Rami moth is much more aggressive than, for example, the Kamehameha butterfly and other caterpillars that feed on Urticaceae. When they are disturbed, they will kind of rear up its head and shake violently. And then that will cause them to regurgitate a green liquid in order to defend themselves from predators. So there are two potential larval lookalikes that also feed on mamaki and other endemic nettles. The first is the red admiral, Vanessa atalanta. This is primarily found on Hawaii Island. It's rarely seen on any of the other islands. And then, of course, our state insect, the Kamehameha butterfly, Vanessa tamamea. This can be found on all the major Hawaiian islands. So this is just a comparison between the three species. The Kamehameha butterfly and the red admiral are both in the same family. They're both nymphalids. So their eggs look much more similar to each other than to the Rami moth. But for early instars, they all show that black head capsule and then the hairy like hairs and spines. So that can be a little confusing during early instars. However, with later instars, they do show a lot of color variation. We believe the color variation is a plastic trait for thermal regulation, depending on where they're located. So the Rami moth has both a black and yellow color morph. From what we've seen so far in our surveys, we can find both colors on Maui and Hawaii Islands. But for the Kamehameha butterfly, this green morph is definitely more common on all the islands. And then the brown morph is found on Hawaii Island, Maui Nui, and Kauai. In terms of the differences between the Rami moth and then the two nymphalids, the Rami moth is larger in size, especially when you're comparing both like fifth instars. The Rami moth is almost double the size of the Kamehameha butterfly. They also have these distinctive red spots that I mentioned earlier on the side of its body, while the Kamehameha butterfly and the red admiral do not. And then in terms of their spines, theirs is very much hair-like, as you can see here, rather than the short, stubby spines and thick spines that the Kamehameha butterfly and the red admiral have. And then for known host plants, there are several genuses in Urticaceae that they're known to feed on. There have been notes of the larva feeding on two other families as well. But what we are most concerned about in Hawaii, in addition to the mamaki that we have seen it feeding on, is two endemic Urticaceae, Acolea, which is also a Bomarea species, and then Olona, which is a Tuchardia species. And then another good way to identify the differences between the Rami moth and the two nymphalids are the feeding damage. So the Rami moth will feed on both young and mature leaves. In early stages, they'll create kind of these holes, these small holes in the middle of the leaves And then as they develop, they create even larger holes right in the middle. And then by the end, they'll completely defoliate the leaves, only leaving these major veins. However, with the Kamehameha butterfly, they actually create, they feed from the leaf margin in. So they'll feed a little circle out of the leaf margin and then clip it and then fold it over to create a shelter or tent-like structure. Then they will feed within the tent until they've run out of food, and then they'll come out and find a new spot to create another tent to feed. And so they'll kind of feed from the leaf margin in, while the Rami moth kind of just walks all around and feeds, creating these large areas of defoliation. And then in terms of distribution, this is all the reportings and sightings that we have of Rami moth so far. They have been found on in residential areas in both potted and planted mamaki, as well as in our native forest feeding on wild Urticaceae. And so we're currently conducting surveys to determine additional locations where this pest may be found. And then to stop the spread, we ask that you do not move any host plants such as mamaki or olona between islands or areas. And then if you do, especially to islands like Oahu and Kauai, where this caterpillar has not been detected yet. And then if you're to move plants, to have them inspected by the plant quarantine branch. And because we are doing active surveys, if you report any sightings, you can email myself. My email is on the slide, or to the plant pest control branch, to Janice. She will also be notified when there are sightings of this caterpillar. And we are in the process of releasing our new pest advisory document through the Department of Agriculture. That should be coming out shortly. And that would be a good resource if you know anyone who are mamaki growers or nursery owners on Hawaii Island or Maui. That would be a good resource for them to look out for and to alert us if they see any sightings on their plants. And so finally, I just want to give a big thank you to the Hawaii Invasive Species Council for funding on this project, to Keahi, he was the first to detect it on Maui, to Jonathan, who was the first to detect it on Hawaii Island, and then to Anna, Irene, and Jaya. They're gracious enough to allow me to survey their areas. And then to Mock, Stacy, and David for helping me with the surveys, to going out into when we are alerted of sightings, to go out and survey the area. And then to Will for all of his photos and expertise on the Kamehameha butterfly. So thank you so much for having me today. I don't know if I'm answering questions now or at the end.
Thank you, Michelle. It's a very good piece of information. So you can answer in the chat box, but we will have question and answer session at the end of the Mark's presentation. In the meantime, I would like to take one group photo. Say cheese. And one more, I think. You can turn on the camera if you like. Group photo. Okay, I can see a few people turning on the camera. Okay, thank you. Okay, this one has lots of, okay. Thank you. So moving on, we will have Mark Wright talking about updates on natural enemies of bark beetle, family Scolytinae, and coffeeberry borer biocontrol research updates in Hawaii. Mark.
Thank you, Roshan. I'm going to switch this over to sharing the screen right now.
Just to introduce, Mark is a professor in entomology and extension specialist. And he does research, all kinds of research from elephant to mosquito. Mark.
Well, yeah. The mosquitoes is a sideline that I don't necessarily do. I have students who do those things for me. But yes, I'm going to talk about scolytine pest today, but primarily I'll talk about coffee biocontrol, coffeeberry borer biocontrol. And, you know, I saw as an option, I could simply run this YouTube video that I'll post the URL for this in the chat, because this is really a neat piece of work, which looks at a broad diversity of bark beetles and their natural enemies in Hawaii, done by David Hansberger, a PhD student in our lab. It's remarkable stuff. So I've put the link in the chats for those who want to check it out. And he's discovered a whole bunch of new species of parasitoids and a whole bunch of new relations with different scolotine beetles that occur in Hawaii, mostly invasive species that he's been looking at. And I think he's going to be the next David Attenborough, by the way. His presentation on that video is remarkable. So I strongly recommend checking that out. But let's just get back to my stuff, actually. The main pests that we have, the main scolotine pests in Hawaii are really pests of crops and a couple of native species. And economically, the most significant are probably those that attack coffee. Black twig borer, Zalesandrus compactus, which we've had in the state for a long, long time. It's extremely prolific species. I think it attacks in excess of 230 different plants, including coffee, and it burrows into the stems. Very hard to manage. More recently, the last 10 years, we've had coffee berry borer in Hawaii. And this is a highly host-specific species. It attacks only coffee, burrows into the coffee fruit, as you probably know. Another one that can be troubling and problematic is the tropical nut borer in macadamia nut. That'll bore into the husk and then into the shell and kernel of macadamia nuts if they're left exposed for too long. If you don't harvest frequently enough, this creature, tropical nut borer, can cause quite significant economic losses. We also have Prolificus shothole borer, Uolacea fornicatus complex. This attacks various crops. We're seeing more and more of this on avocados. I think it goes after macadamia as well, particularly if they're stressed. In fact, we have a couple of species, at least 2 species of Uolacea in the islands. And actually, David, whose work I just showed in the previous slide and recommended you check out the YouTube video, has just discovered the very first parasitoid of Uolacea ever in the world, which is a real neat discovery. And I think he has some video of it in that YouTube show. There are a couple of species that attack native trees as well. So vectors of Rapidohea death, for example, and black twig borer attacks native trees like koa, for instance. But I'm going to speak mostly about coffee, and specifically about coffee berry borer, because it's economically so important, and because it's the one that we have the most data on with regard to biocontrol at this point. Honestly, the only one. Just a little about coffee, and you guys probably all know this, but we produce a huge amount of coffee actually for such a small place. 38 million pounds back in 2016. That's a lot of coffee. It's the only state in the US that produces coffee. Puerto Rico has an industry as well. It's a tiny bit in California. So it's a hugely important industry for Hawaii, particularly Kona coffee has this real boutique attraction. KR coffee is excellent. Kauai Coffee Farm is the biggest in the world, in fact. Back in 2010, we were unfortunate enough to have Hypothenemus hampii, the coffee berry borer, invade Hawaii. Originally an African species, it's now present in almost every place in the world that grows coffee, and it's coffee's worst insect pest worldwide. Over here, as soon as it became established, it started causing pretty severe impacts. There was significant yield losses, massive yield losses in the beginning. And this also drove farmers to change their crop management style. Previously, they could fairly easily get away with not doing too much pest management, except black twig borer sometimes and a few other things. Coffee berry borer changed all that, and the growers had to modify their practices. There's some real problematic aspects with this creature in terms of managing it. This is true for most scolotines, for most bark borers, bark beetles. They complete their entire life cycle within the plant. Coffee berry borer completes the life, aside from the female flying between plants to find new places to oviposit, the whole life cycle is completed within the fruit. There can be multiple generations produced within a single fruit. And of course, they're decimated in the process. To successfully control this creature, sanitation is super important, which means clear picking berries and picking them off the ground so that you don't have a reservoir of the pest out there for the next season. That's not easy to do here. We have labor cost issues, and we have uneven land issues. Doing clear picking on lava flow is not an easy thing to manage. We also have feral coffee and unmanaged coffee that serve as reservoirs out there. And these are all problematic issues. You also have to monitor this thing to make management decisions. It's a labor intensive. It's a mission to control this pest successfully. So what have our responses been? What's happening right now? The immediate response when the invasion was first detected, of course, was to look straight into trapping, to monitor this thing and to try and suppress it. We've looked more and more at ways to sample in an economic manner that you can actually monitor damage and infestation levels and make management decisions. I've just mentioned sanitation. That's something that was immediately recommended and applied as far as possible. We also have really few pesticides labeled. There are 2 or 3 compounds that are labeled for use on coffee, and that's problematic. And many farmers don't want to use synthetic pesticides either. We have a couple of biological options. Beauveria bassiana is a entomopathogenic fungus that is quite effective against coffee berry borer if used frequently enough. I'll show some data shortly on its efficacy. There's some predatory beetles that prey on the CBB larvae and pupae, and then we're now looking at parasitoids, which I'll speak about for the most of this talk, actually. Currently, these are things that we're actually researching, culminating with classical biocontrol using Phymastechus coffeae. We're considering other things right now, like looking at repellents that may have some potential to keep CBB away from plants, and we're getting some success with verbenone. It's an anti-aggregation pheromone that scolotines produce. We've been looking at Beauveria bassiana effectiveness and the impact that shade has on how effective it is, and that work's been done in Puerto Rico, actually, by a PhD student who's just finished up with me. But again, I'm going to concentrate mostly today on Phymastechus coffeae, which is a little parasitic wasp from Africa. Let's look quickly, though, at the effectiveness of Beauveria bassiana. These graphs show the results of trials that we conducted in Kona at low elevation, moderate elevation, and high elevation, and the elevations are shown over there in the panel on the right. And we also had 3 different doses. We applied a high dose, a low dose, and a medium dose, which are the doses on the label: 8, 24, 32 fluid ounce per acre. And of course, in the experiments, we had an untreated control and a silicone control, because you include silicone with with Beauveria when you spray it as a spreader. And then the y-axis shows percent infested berries, and we detected this as CBB with fungus. So it's really apparent mortality, as we'd call it in biocontrol research. And what you see is at low elevation, Beauveria was significantly less effective. You get maybe 15% mortality, and this is with repeated applications, monthly applications. At moderate and high elevation, you get, let's say, about 24% mortality of CBB, substantially and significantly higher than the controls in the silicone, and also significantly higher than at low elevation, probably a temperature difference effect. If you make repeated applications, which are expensive, you can achieve quite good control of CBB over the course of a season with Beauveria, but it's expensive. You have to keep it up, and you have to ensure very effective coverage of the plants. So while it's effective, it's expensive, and it's logistically difficult. Some of my colleagues, Peter Follett and Andrea Kawabata, and a couple of others have been looking at the potential for using predators that occur here as augmentative biocontrol agents, meaning that we'd go out and release them into the field as needed, like a living pesticide. And they found that these square-neck grain beetles, Cathartus, they do go into CBB tunnels, and they do feed on the beetles, and they do eat a substantial proportion of the population, you can easily rear them in crushed corn, as you see down there in the image. And they can supply these starter kits to farmers who can then grow them up and release them into the field. So they may play a role in suppressing the populations. We don't know yet exactly how effective they are at doing that. They don't prevent damage to the berry, though. That's the problem. So what we're interested in doing is looking at classical biocontrol, which means we release a biocontrol agent here that becomes established and sustains itself in the field and provides us with long-term control of the pest, or potentially augmentative release. Again, mass rearing a natural enemy and releasing it like a living insecticide. And the species we're looking at is called Phymasticus coffeae. It's a yellow-footed wasp, which is a little parasitic wasp originally from Africa. I believe it was collected from Uganda and Kenya, which is part of the original provenance of coffee, not necessarily coffee Arabica, but coffee in the general sense. And it attacks coffee berry borer there. CBB is not a problem in Africa, probably the result of multiple natural enemies that occur there. It's a tiny little thing. It's about the size of a trichogramma, for those of you who know and care about trichogramma. Have a relatively long life cycle within the host, at a short adult life cycle. I think they live for 2 or 3 days as adults, but up to 3 or 4 weeks as larvae inside the host. So in experimental work, people have shown that you can get in excess of 95% parasitism, which is impressive. And it's supposed to reduce damage out in the field. That's been tested out in Colombia and 1 or 2 other countries where they grow coffee. But in those places, it didn't seem an excellent candidate for classical biocontrol, mostly because they clear pick all the coffee every year, so you don't have a reservoir that can sustain the parasitoids. The image down the bottom there shows a great little shot of a CBB female adult with the Phymasticus emerging from the rear of her abdomen, where it's previously its parent previously it parasitized the adult. The female and the male developed inside, and then they emerged through a little hole at the rear of the beetle to go and seek out further victims. All right, so we're looking at the potential for classical and maybe augmentative use in Hawaii. Of course, one of the main concerns that we have is non-target impact. And it's one of those things that we look at very carefully when considering bringing in a new biocontrol agent to Hawaii. We have about maybe a few more than 20 scolotine species in Hawaii that are natives. They're in the genus Xyloborus. There's maybe one in the Hypothenemus genus, but probably not. That's probably a misnomer from ages ago when people made an error and recorded a Hypothenemus as native. So we're probably dealing with a group of Xyloborus species, which are in a different subfamily, a different group to Hypothenemus. But we're concerned whether Phymasticus coffeae would pose any threat to these. Typically, insects that are parasites of organisms that live within plants are somewhat polyphagous. They'll attack a range of hosts, as opposed to being monophagous, which is what we really would prefer for a biocontrol agent. So there's a bit of a concern. All right, so the work that we did for the last couple of years examined whether Phymasticus will attack native species and will it attack other pest scolotines that we have here. A big concern here was where the endemic species are. People don't know. They haven't been looking for these tiny, insignificant little inhabitants of decaying wood. So we employed a postdoc, Conrad Gillette, who went out often with David and others to go and look for the endemic species so that we could know where they are, what they're utilizing, and put them into our non-target screening experiments in quarantine, which was done at Volcano with the Forest Service in Tracy Johnson's facility. So this involved surveying for endemic scolotines, going all over all the islands, sampling a whole range of different vegetations, whether they were introduced plants, native plants, and so on, looking for endemic as well as introduced scolotines. And Conrad and company would use traps that they'd put out to find the beetles, and they'd often collect them directly from wood. We'd often be able to obtain live beetles that way that could be used immediately for non-target screening in the quarantine. These are some of the locations, and I guess these are the benefits of being a postdoc or a student, is you still get to go and do these things while old people like me sit around writing grants. This is on Molokai. You can see they were up here in forest. They're hanging out a Lindgren trap, which will then attract scolopendras, and I'll dig them out later and check them out. There were some remarkable findings from this work, as an aside almost really, but rediscovering species of native scolopendras that haven't been seen for decades. I think this species had not been seen for 50 years prior to Conrad and company finding it over on Molokai. And then they were able to locate a number of endemics, like, for example, this little one from Maui, which I think also had not been seen for a good number of years. I didn't think the host plant was known. Another native from Molokai. And bear in mind, these are tiny insects. They're a couple of millimeters in length, and they occur inside of wood in most cases. So cryptic, really unknown and unseen. And we, thanks to the efforts of Conrad and others, able to test a good number of them in our non-target screening. So the way that this was done was you would put the potential non-target and compare that with coffee berry borer as a positive control in Petri dishes in the quarantine, and then introduce the parasitoids, the parasitic wasps, into those containers and see if they were able to parasitize the beetles, if they were able to develop in them, and if adults were able to emerge eventually. Ultimately, we were able to screen, in fact, I think that should be 43. This is, I had not updated that. I think it's 43 species were finally screened, including 6 hypothenomous species. Those are introduced. A bunch, 7 endemic species, some other cuculionids, other weevils that occur in Hawaii, and then a number of other things. For example, coccinellids, which are beneficials, sylvanids, nitidulids, which are just other things that we could expose them to get some idea of their host range and ability to parasitize things that are not coffee berry borer. This is quite a lengthy process. We'd have to take 6 to 8 weeks to do it because of the long larval life cycle of these insects. And then after that period, if nothing had emerged, the insect, the potential host would be dissected to look for parasitoids. All right, so among other things, we also looked at behavior of the wasps when they were exposed to these different parasitoids, beetles. But the main measure that we were interested in was, were they able to parasitize them? If so, how many did they parasitize, and were they able to survive, survive, and reach adulthood inside those non-target hosts? What I'm showing here is some really summary results. It's just a few of the species. We have a paper in press right now that has the full range of species that we looked at. If you look at Hypothenemus hampii, coffee berry borer, we had almost 80% parasitism, about 70% emergence. Other Hypothenemus species, Hypothenemus obscurus, 50% parasitism and about 40% emergence. And I'll show something more interesting about Hypothenemus in a moment. If we look at other species, like Xyloborus, for example, 0% parasitism. None of the exotic scolopendras were attacked. Not a single one of the endemic, the native species was attacked. The parasitoids didn't even show an interest in trying to parasitize them, and nothing else that we exposed them to. So they show specificity at the genus level, it would appear, and they appear to be way more effective on Hypothenemus hampii than others. What you see in this graph is the different Hypothenemus species that have been introduced into Hawaii. Hypothenemus hampii being CBB, obscurus, the tropical nut borer, etc., other species that are here. And this graph shows the percentage parasitism in the white bars and the percentage emergence of adult wasps subsequent to parasitism for each. You'll see Hypothenemus hampii had the highest parasitism and emergence, and the others significantly less, down to, in fact, 0 in Hypothenemus eruditus. If we look at the phylogeny, the evolutionary history of those Hypothenemus species, and I've just kind of inferred it from this phylogeny published by Johnson and company back in 2018. I've extracted the species we have and redrawn that phylogeny here. You'll see that the more distantly related phylogenetically that the beetles are to Hypothenemus hampii, the less likely they were to be considered to be hosts, and in fact were not utilized as hosts in the case of Hypothenemus eruditus whatsoever. So it would appear that phylogenetic relatedness plays a significant role in driving specificity. Hypothenemus hampii is the only scolotine in the world, I believe, that's completely monophagous, and it has adaptations to toxins. It has endosymbionts that allow it to survive toxins that coffee subjects it to. So I think those have posed some additional evolutionary constraints on the parasitoids, and has driven the preference and the effectiveness of Phymasticus on coffee berry borer. Okay, so sorry. What we see is reasonable specificity in Hypothenemus. I suspect strongly that if we put these out in the field, that you're going to see that they will only attack Hypothenemus hampii and perhaps a few of the others, because there'll be plant-associated cues that go along with attacking coffee berry borer, cues that are provided by the coffee plant itself as it's being attacked, where the other species will not be providing those cues. What is extremely encouraging for us is the fact that there was zero parasitism of those other Scarletinae species that we exposed them to. They would not even consider them as potential hosts, which gives us confidence that making that release into the environment would be safe. We wouldn't be impacting native species. And currently we have a petition in for release. It's with USDA, APHIS, and I think a revision is being sent to the Hawaii Department of Ag very, very shortly. So hopefully one of the days we can get this insect out into the field and start to see if it impacts coffee berry borer. All right, and there's just some acknowledgements. USDA and HDOA provided funding. Quarantine facilities over in Volcano were a lifesaver. And we have a bunch of collaborators all over the world, in fact, who produced wasps and shipped them to us, and provided all kinds of other support. And these guys are all co-authors on our paper, which is in press now at Journal of Pest Management. If anyone's interested, give me a shout and I can send you a copy soon. All right, that's the end of the presentation. And I'll be happy to answer any questions if you have. Thanks,
Mark. I missed one point, I think. So the Phymasticus coffeei is what kind? Did parasitize larvae?
Oh no, it parasitizes the adult.
Okay.
The adult beetle.
Mm-hmm.
Let me just go back and show you the very.
So that lay eggs on the adult. That means.
So if you look at this, sorry, I'm not sharing my screen now, am I?
No.
Hang 10 coming up.
I saw one photo of the adult coming out from the
right.
The parasitoid coming out from the adult. Yes.
Right, but okay. All I have here is a sketch of it actually, but. As the CBB is burrowing into the bean, so the parasitoid goes and sits on the abdomen, like you see in that image there, and she inserts her ovipositor between the elytra and into the abdomen, and then she'll lay 2 eggs, a male and a female egg. And then eventually they emerge, like you see.
Okay, got it. Yes, got it. I saw that. Okay, thank you. Questions for Mark as well as Michelle?
Yeah, Roshan, I've got a question for Mark.
Yeah, go ahead.
Mark, aloha. On farms like Kauai Coffee, where we don't have nearly enough labor to do the clean strip picking, you mentioned a likelihood of achieving parasitism. Is that what I say that right? Is that possible or is that likely?
I'm sorry, so the likelihood of achieving parasitism?
Correct, on farms that aren't able to strip pick, meaning we'll almost always have some.
Oh, I see. Right. So the sort of curious benefit of not having complete sanitation is that you maintain a reservoir of the parasitoids in the field. So in other words, from season to season, you're sustaining the parasites instead of removing them completely from the field. That's the kind of silver lining of not completely sanitizing. And I think that's what I was alluding to, is that while classical biocontrol using Phymasticus didn't work in Central America and so on, because they clear pick, it's more likely to work here because we have a reservoir year round.
Yep. And farms just don't take care of their.
Well, I mean, I know your farm is huge, right? It is impossible. Kona farms, it's really difficult to remove everything. I know for sure.
Thank you. That's great work. Appreciate it.
Most of my colleagues. I see there are some questions in the there's one for Michelle there. Oh, okay. I see Cheng is asking, what is the largest ag industry in Hawaii?
It's there, I think.
Say again.
Somebody answered already. Seed corn.
Seed corn, right? Yeah.
Seed corn, and second is the coffee. So yeah.
There we go.
Any more questions for Mark and Michelle? Michelle, do you know where did you find Rami moth in the Big Island? I think Franny has a questions. Mm-hmm.
So it's in the Lower Puna area. Do you want me to pull up the map again that I showed the distribution earlier?
Yeah.
Okay, let me reshare my screen. Thank you. Yeah, so currently it's in the Lower Puna Pahoa region. We did survey in Volcano and did find a few caterpillars up. I know along the roads there are a lot of mamaki there. And then Mountain View was where it was first reported.
Thanks, Michelle. So, questions?
So just to comment here, Michelle is actually interested in looking at other potential hosts of that moth in Hawaii, and there are a bunch of native species right in the family. So it would be really useful if people who know populations of some of those really scarce things, if they could let us know where to find them. That might be helpful.
Yeah, that would be great.
So, Mark, do you know when we will be able to release a parasitoid for coffee berry borer? Any estimated date?
Yeah, it's kind of like asking how long is a piece of string. Yeah. Well, I mean, it can take ages, as you know, for biocontrol agents to get out of quarantine. So right now well, Peter and Fazila actually wrote up the petition and submitted it to APHIS, and they've sent it back with comments. We also have to complete a cultural impact assessment still to include into the petition, which should be done within about 2 months, I think. So I'm hoping within 2 months we can have everything back in motion. And from what I've seen previously, it can take, well, at least a year, if you're optimistic and really lucky.
Okay.
I know with Erythrina gallwasp, for instance, we got that out really, really quickly. Well, HDOA did. Maybe we can get lucky here again, but certainly those.
Hey, Mark, this is Stephanie.
Hey, I can't hear you. Did you?
Maybe you muted again, Stephanie.
Yeah, it's my stupid connection. Sorry. Just wondering that the only fear of this biocontrol attacking non-targets would be really rare and hardly ever found native scalidid beetles who don't seem to have any advocates anyway. I mean, is that the barrier? I mean, would that, I mean, that wouldn't, yeah, I'm just curious.
That's the concern that we had, was that we'd introduce something that might attack an endemic species. And yeah, you're right, they're obscure, and perhaps no one other than us would really care about it.
Yeah.
And we've completed that work. We've shown that that's a non-risk. So I don't see that as a barrier. So there's a number of bureaucratic steps that just trip you up along the way with the hearings and getting permission granted to make the release that can be troubling. So, you know, I mean, the Board of Agriculture or someone could say, no, we haven't looked at enough native species yet. We need to include more. And there you go, you're held up. And that's happened with other cases. I'm hoping it doesn't happen here, because this is very clear cut, I think, that this thing is safe. So I don't foresee
It's hard to even find, it's like hard to even find the native ones to test them on, you know?
No, correct.
Anyway.
I mean, you know, we actually made a substantial effort to do that. And again, I think we've shown very conclusively that they don't attack any of the native species. So I don't foresee that being an issue. I'm optimistic. Sometimes at my peril.
Hey, Mark, I'm just curious if you connected with Kyle Roy and those guys over at USGS for the beetle work they're doing with the ambrosia beetles on rod. Do you know if it affects any of the ambrosia beetles that are thought to be facilitating the spread of rod?
It does not. It doesn't attack any of those species at all. No. We've just found actually a new species that's going to be described soon that attacks some of those beetles. But no, Phimesticus coffeae does not.
Thank you.